This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 38 for June 25th, 2009. Hello, everybody. I'm Vincent Racaniello, and it's time for TWiV, and my guest today is Glenn Rall. Welcome to TWiV, Glenn.
Thanks, Vincent.
Glenn is a member in the Basic Science Division of Fox Chase Cancer Center in Philadelphia. He's also the opinions editor at PLOS Pathogens. His laboratory works on Measles Virus, and that will be today's topic. Alan Dove could not make it today, and Dick DePalmier is somewhere in the world. We're not sure where he is. So it's you and I, Glenn. I'm sure we can carry the TWiV banner together.
I suspect we can. We'll keep it going for an hour for sure.
Our first story is very interesting. Virus suspected in carp die-off. Arizona Game and Fish Department biologists believe a virus that killed thousands of carp in Lake Mojave is doing the same at Lake Havasu. And this is in Kingman, Arizona.
Mm-hmm.
Carp, as you may know, Glenn, is a food fish for much of the world. So the supply is being threatened. And of course, koi are ornamental fish that people have as pets, and they often die of this koi herpes virus. So it's a very contagious infection. The herpes viruses, of course, are large enveloped DNA viruses.
Right.
So this is a typical herpes virus. There's quite a bit of research actually going on these viruses. Apparently I think the first discovery of this virus was in the 1990s, yes, in England in 1996. And since then, they've been found all over the world. The fish die, basically. They get sunken eyes and swollen gills. And if you have pet fish, of course, this is a problem. Apparently, some of these ornamental koi go for as much as $100,000.
You know what? I actually looked at this story as well, and you know what I thought was interesting is that goldfish, which are in the same family, are not affected by this virus. So it's extraordinarily specific.
Yeah, that's very interesting. Possibly receptors or something else. We don't know. But in the case of this article that I pulled up, it's in a lake somewhere, and the carp died. And they said in the last sentence they knew it was this virus because it's the only species in the lake that died. And in this case, the infection came from a lake farther upstream, so this virus often spreads through connected waterways.
Mm-hmm.
And I would guess that it probably also spreads in boats. If you carry a boat from one lake to another, you have some water in it, which often happens. You can carry viruses. Last summer, I was canoeing up in the Adirondacks, and we would do some portaging from lake to lake. And there were these signs that said, don't bring any water from one lake to another.
Huh.
And they weren't about viruses. They were about various algae and snails and so forth. But viruses could be transported in the same way.
Right, and probably actually as a herpes virus, it probably has a fairly long lifespan in absent cells in water. So that makes some sense.
Yeah, and in fact, there's some indication that these viruses might have a latent phase also in the fish.
Right.
So you could bring a fish home, and it looks fine for a while, and then the virus is reactivated. And in the same way, you could stock fish in a stream or a lake and unknowingly put infected fish in there.
Well, didn't they imply that this might be even a temperature-dependent phenomenon, so that as the waters get warmer, this is what causes the virus to in a sense, reemerge and cause the sickness in the carp, and then during the cooler periods, the virus may be quiescent or latent?
Yeah, absolutely. There's some range of temperatures where it replicates, and below or above that, I think somewhere in the 70s,
right.
It didn't grow. So yeah, as the temperatures change, so it may be in the springtime as things warm up, you can have outbreaks. And very high mortality, 80 to 100% mortality in susceptible populations. Here we go. Clinical signs of disease most commonly expressed when water temperatures are between 72 and 81 degrees.
Right.
And I have a picture here of a koi with mottled gills and shrunken eyes. So there's actually quite a bit of research being done on these viruses because they are agriculturally important. And there are lots of Journal of Virology papers The sequences of many of the genomes have been done. And I also found one paper whose title is "The Major Portal of Entry of Koi Herpes Virus is the Skin." So there's some controversy over how these viruses get into fish. A lot of people assume that it was the gills. And in this paper, they say, no, it's the skin. And then I also found other papers suggesting there was a latent phase. So there's quite a bit of research here. It's very interesting, in very interesting journals with unusual names that we're not used to. But we'll put a couple of links in the show notes. I just wanted to point this out. It's quite interesting. And you see these stories now and then in the press of fish die-offs from various viruses, not just this one. But this is a herpes virus. It's quite an interesting one, and apparently increasingly globally important. It's considered an emerging infection of fish. Our next story, a new influenza vaccine being developed by a company called Protein Sciences. And this was an announcement from the Department of Health and Human Services on June 23rd. HHS announces advanced development contract for new way to make flu vaccine. So this is a small company in Connecticut, and their approach is basically to express the viral hemagglutinin of influenza virus in insect cells using baculovirus vectors. So this is a new approach to flu vaccines, and they're giving this company, the HHS is giving this company $35 million to develop this vaccine. It has to be tested and so forth. It hasn't even undergone a clinical trial, but they say in one of these articles that the FDA, of course, could bypass all that if they said, let's use it. But I can't imagine not having any clinical data at all from such a vaccine.
You know what actually kind of surprised me about this was how long it's taken us to get to this point, because the generation of the flu vaccines is so arduous, still, taking lots of time growing them in eggs. So to come up with a baculovirus-based approach makes terrific sense, especially if you can find a fairly cross-protective hemagglutinin antigen to express.
Yeah, I think part of the problem is people have tried hemagglutinin subunit vaccines in the past, and they're just not terribly antigenic. They don't make a good immune response. The best adjuvant appears to be the virus particle, which makes perfect sense, of course. But the current killed or inactivated flu vaccine is whole virus, and it's very immunogenic. If you take out the HA only, it's not as good. So they may have the same issue here. So we'll have to see how they get around it. My guess, if they put this into people in a clinical trial, they're going to see low levels of neutralizing antibody production. But we'll see. I don't think we've ever talked about baculoviruses on TWiV. These are insect viruses. They're large DNA-containing viruses, very interesting in their own right, but they happen to be great vectors for synthesizing proteins. You can clone genes into their genomes pretty easily, and then you infect insect cells with the recombinant viruses, and they make a lot of protein. So that's the approach being used here. I don't know how they're purifying the HA. They don't provide any information on that. Do you know anything about this?
No, I don't. And do you have any sense of which hemagglutinin they're going to be using for this candidate vaccine?
No, other than it's not, one of the 2009 ones. I don't know which exact one they've decided to use, but they will express the protein in these cells, and then it has to be purified. Of course, I don't know how they're going to do that, and then they have to inject people, and they'll have to do some trials to just show that it's safe and generates immunity. But they said so far they've made a few million doses. I mean, the obvious advantage of this is clear. You can make a vaccine relatively quickly. You don't need to make an egg-adapted virus. You don't even have to work with infectious virus. You just work from the sequence of the HA protein. So there are lots of advantages, but the disadvantages maybe it doesn't work.
Right, exactly. Well, it certainly is timely given all of what's happening with the H1N1. I mean, I just got my most recent issue of Science has actually a picture of apparently patient 0 on the front cover.
Is that right?
This
little boy in
guy from Mexico. But interestingly, actually, I thought there was also an article that was in Science that I was amazed at how quick the turnaround must have been, because this is actually lots and lots of data on the pandemic thus far. And one of the positive points about this, or I guess optimistic points about this, is that it appears that the clinical severity for the H1N1 viruses are fairly low. The case fatality rate seems to be only about 0.4%, so that's substantially less than the 1918 pandemic that everyone is disfavorably comparing this to.
In fact, if you look at the CDC site, they have these graphs of pneumonia, influenza, excess mortality, and this season there is no excess mortality at all. Compared to last 2 seasons ago, I believe there was a excess mortality over the epidemic threshold, as they call it. So it is quite mild. It's almost like seasonal flu, although people are dying, and we know because every death is documented in the press quite well. But it is quite mild. It's very interesting how much research is being stimulated by it, which is good, and they're spending a lot of money to do this. So this is one example. Maybe this vaccine wouldn't be pushed as hard as it is now, if it weren't for
had it not been for the pandemic. Yeah.
Now, do you know of any human viral vaccine that is made in insect cells yet?
No, I don't.
Yeah, I don't either.
I don't believe that any are. So perhaps, I mean, maybe this is in a sense the most relevant virus to try, but to test whether the baculovirus approach is feasible. You can generate a higher number of doses, turnaround time for this. I'm unaware of any other cases in which baculo is used as a sort of basis for vaccine development.
Most of our vaccines our human vaccines are pretty old technology.
Yeah, well, we'll talk about that in a little bit. We're pertinent to measles virus, but the vaccine that was developed in the early 1960s is still the one that's in use today.
I suppose at one point in the future that will change, but these things change slowly, especially when you have a vaccine that's effective. It's very hard to change it, but in the case of flu, where you have to make one every year, alternatives are great, especially if they're more rapid.
Right.
Okay, so that's the flu vaccine from Protein Sciences, and we'll put a link to that in the show notes. And we have a couple more stories, but before we get to them, it's time for a word from our friends at Citrix, the makers of GoToMyPC.com. And you know that more people these days are web commuting. They stay home 1 day a week, and they access their office PCs from home. And people love doing that for obvious reasons. You don't get stuck in traffic. You get to work in the comfort of your home. You don't have to get dressed even. And so if you can do that, it's great. And that's why I recommend GoToMyPC, which is brought to you by Citrix. It's the best way to web commute. GoToMyPC is the easiest remote access service on the market. You simply log on to GoToMyPC.com, and your office desktop instantly appears. You can use any program, work on any file, check your email, access network resources, anything at all. It all works. And you can use GoToMyPC to work from home even 1 day a week, and you'll spend less time in rush hour traffic and more time being productive. So you can try this wonderful program free for 30 days, but you must visit GoToMyPC.com/podcast. That's GoToMyPC.com/podcast for a free 30-day trial. You have a long commute, Glenn?
No, I'm really fortunate. So Fox Chase is in Philadelphia technically, but it's in Northeast Philadelphia. So it's out of Center City, and we live only about 5 minutes away from the cancer center. So my commute is quite a breeze.
Do you drive in?
I do. There are times I could bike in or walk in pretty easily as well. But for a while, I would have to drive just so I could pick up my kids from daycare. But it's extraordinarily convenient.
That's great.
How long is your commute's pretty substantial, I suspect, right?
I'm 38 miles away from my lab. So in the best of times, it's an hour or 50 minutes, but there are rarely the best of times as we're talking about crossing the George Washington Bridge. So yes. I try to work from home as much as I can, but for one reason or another, especially when I'm doing experiments, it's not possible. You can't just take a day off.
Right, but your point is right, that if you have the opportunity to work from home on a grant or on papers, being able to access all the information that's stored in your office computer is absolutely critical. Yeah. So
my office computer has a firewall. In fact, all the computers here are firewalled, and you can't access them unless the university pokes a hole in the firewall for you, which is very hard to get permission to have done. So a program like GoToMyPC makes it easy. You don't have to break the firewall at all. It just works. If I do get to stay home, I use it. Our next story is a man being treated for rabies after the bite of a rabid raccoon. And This is a story from ProMed Mail, and we've talked a lot about rabies on TWiV. I thought this would be a good story to bring up just to warn everyone that you have to be careful. This is a man who was bitten by a rabid raccoon. He's 29 years old, bitten on the right leg while working on a porch at home. He said he was busy working and never saw the animal approaching until it lunged at him and bit his leg. And he fought off the raccoon and killed it, and they took the head and brought it in, and it turned out to be rabid. So he's getting rabies vaccine, which will prevent him from dying, essentially. And you have enough time to inoculate someone after the bite. And so, if you're working in a rural area, you just have to be careful, because animals who get rabies can behave abnormally. So this raccoons normally don't attack people, but when they're rabid, they do.
That was actually pretty wise of him to have been able to kill the raccoon and then bring it in for tests, because time obviously for him was of the essence to be able to get the vaccine in so it could actually do some good for him.
Yeah, absolutely. And many times you don't get the animals, and then you have to just immunize anyway.
Right.
And so sometimes people will touch bats or be in a cave with bats and not get the animals, and you don't have that luxury. So he was very smart. So if you are bitten by an animal in the wild or even near your home and it seemed to be behaving oddly, you should try and get it if you can. Although I don't know if I would fight off the raccoon and kill it. I just would kick it and get rid of it, I suppose.
That's right. That's what I meant.
Anyway, it says in this article, rabies is most commonly found in raccoons, skunks, and foxes. Although dogs, cats, horses, cattle, bats, and bobcats can also be infected. And last week we read an article from an email from Jim who talked about skunks being under his neighbor's front porch, and so they can be infected. The disease is extremely rare in small rodents, rats and mice, squirrels, opossums, and in rabbits and hares. The other day I was driving in, and there was an opossum on the side of the road. He just kept walking into the road, then turning around, then walking into the middle of the road and turning around. And he's behaving really oddly. I wonder if he was rabid. And this was messing up traffic, of course, because everyone was stopping for the animal.
Maybe he was just frustrated by the commute into New York City.
It could be. You don't see opossums out very often, They tend to hide, so that was unusual. So rabies, of course, is an RNA virus, enveloped single negative strand RNA. Very similar to the virus that we'll be talking about today. Different family, but overall similar structures, wouldn't you say?
Yes, indeed.
And rabies is a killer, almost 100%, basically 100% fatal. There are just 2 cases of people who have survived without being immunized, and these are people that received the Wisconsin protocol. They put you in a coma and chill you, and it seems to help survival. But I remember Hillary Kaprowski, who's down in your neck of the woods, Glenn, talking about rabies here a couple of years ago and saying basically you're dead unless you get the vaccine, or if you don't, try and get put in a Wisconsin protocol.
So the Wisconsin protocol is just the principal objective of it is just to put you on ice, in a sense?
In a coma, yeah, I suppose. It was first done in Wisconsin. That's why it has the name.
So, and it's presumed that by putting you in a coma it just decreases the neurotransmissibility of the virus?
That would be an assumption. They don't really understand, because it's only been done twice, I believe, in people.
Huh.
And they haven't studied it in an animal. But that would be the idea, that the transmission would be inhibited, yeah. And that's a good point. This man was bitten by this raccoon on the leg, and so the virus has to make its way into the central nervous system. It takes time, and you can interrupt that with immunization or perhaps by putting you in a coma and chilling you. I don't know what the mechanism is. It's a good question.
I know that actually, I think it was a couple of years ago, there was a fairly substantial number of raccoons, in fact, that were found at least on the East Coast to have rabies virus. That's been apparently that has been sort of slowly but inexorably spreading further and further west.
Yes. I mean, there was a time in the U.S. when we thought it was gone, and then it came back in the form of raccoons. And in fact, even in New York City, there were some rabies-positive raccoons. So I think the assumption is that the wild animals have it. There's not much you can do about it. Of course, in Europe, they do immunize the wild animals. They put bait containing a rabies vaccine in the wild. And I'm not sure if it's done here. I've forgotten if that's done in the U.S., but that can be helpful in curtailing the amount of virus in the wild. But in the U.S., when all the pets are immunized, the pet dog rabies is not much of an issue as long as they're pet dogs and immunized. Of course, there are lots of wild dogs here in New York City, and they're not immunized, so you have to be very careful. They roam the streets late at night. I remember years ago when I lived in New York, I was driving home about 3 a.m., and there were packs of dogs just wandering the streets. Hundreds.
Really?
Just wild
So
dogs.
feral dogs in New York
Yeah,
City?
I mean, I presume they were wild because they're running around and they looked wild to me. But they don't do that during the day. They're off somewhere. I don't know where, but it was very weird.
We have odd things in Philadelphia, but I don't believe we've ever seen packs of wild dogs wandering around. But
So they could be dangerous. They could have rabies. They could get it from a raccoon in Central Park, for example. But
right. Well, I mean, this guy was just working on his porch, and this thing, obviously rather aggressively came up and attacked him. So it doesn't even necessitate that you're, out deep in the woods. These things will come and find you if they choose.
Yeah, I don't know where this was. Could be UK. Oh, no, it's in North Carolina.
Oh.
Population 11,000, so it's quite small.
Pretty rural then.
So you have to just watch out, everyone. With the warm weather, everyone being outside, be careful. Keep an eye out. Our last story is a way of segueing into measles, and this concerns the outbreak of measles in Wales, which has been going on for quite a while, right, Glenn?
That's right. Yes, indeed. This is the number of cases of measles virus has been increasing actually pretty much worldwide, particularly in the UK. So England, and it's been increasing in the United States, and Wales has had a problem probably for the last few years. The rates of measles virus have been increasing, but it's particularly notable this year. Since the beginning of 2009, there have been almost 300 cases that have been reported, and of those 300 cases, this resulted in approximately 34 hospitalizations. So, about 12% of the acute measles cases, and obviously this is in a part of the world where there's good medical care that's available for any of the sequelae that may occur following measles infection. So it's a pretty dramatic consequence of infection, and the hospitalizations is a pretty high rate.
These are presumably in kids mainly, right?
Yeah, I mean, I suspect most of the cases, whether they're in Wales or in the United States, all of these, or virtually all of these, are infections in children under 5 years of age, and virtually all of them are in kids who have not been vaccinated.
So these are not vaccinated for one reason or the other, which we'll probably get into in a few minutes, right?
Yep.
So we'll talk about measles. Before we do, let's have a word from our friends at the American Society for Microbiology.
Now available in 2 conveniently sized volumes, Principles of Virology 3rd edition is completely revised and updated to reflect important advances in the field. This textbook continues to fill the gap between introductory text and advanced reviews of major virus families. 2 volumes provide upper-level undergraduate students, graduate students, and medical students with a state-of-the-art introduction to all aspects of virology. Visit ASM Press at estore.asm.org to view sample chapters and the complete table of contents. Request your examination copy today at estore.asm.org.
Okay, let's talk about measles, Glenn. Where should we start?
Well, maybe we should start with the assumption that I suspect a lot of your listeners assume that measles is kind of a virus of the past. When we think about viruses that threaten us today, ones that we're concerned about, obviously influenza virus comes to mind, maybe West Nile virus, other viruses that have, particularly graphic or notable consequences like Ebola virus. These are probably viruses that most people would say that they fear. And then, of course, viruses like HIV. But measles virus, I think, is often lumped in with a variety of other pathogens that have been considered to be childhood rites of passage that happened some decades ago that no longer really are having a substantial impact on the human population. And while that's understandable, especially in countries like the United States or most of the countries in Europe, because the rates of measles virus as a consequence of vaccination are indeed quite low, throughout the entire world, it is still remarkable how many individuals are acutely infected with measles virus each year and what kind of impact that has on human mortality. So obviously it's difficult to estimate or to get a very accurate number of the prevalence of measles virus worldwide, but it's thought that there are approximately 20 million infections, acute infections, that occur each year. And of those 20 million, approximately 500,000 individuals die as a consequence of the infection itself. So depending upon where you look within the world, measles virus still remains a very, very substantial human infection.
So all of those 20 million infections are preventable.
Right. Oh, yeah. I mean, this is a virtually 100% preventable illness. In fact, one of the challenges of the vaccine, while this vaccine, and we will probably talk about this more fairly soon, but while the vaccine is extremely efficacious, it is a live attenuated virus. So this means that the virus or the vaccine, when it is delivered to the individual, replicates, albeit at a much slower rate. That replication induces a potent immune response that then affords protection should you sometime down the road encounter the real replicating pathogen. But one of the requirements of any live attenuated vaccine is simply that it must be kept cold. And this is called preserving the cold chain. And for delivery of vaccines in developing regions of the world, that's a huge challenge. And if the virus is allowed to increase in temperature to room temperature or higher, the virus is effectively killed and no longer can afford the same level of protection that it once was able to when it was, prior to being inactivated.
So how can you tell if someone has measles? Is this an easy disease to diagnose?
Well, I mean, there are certainly hallmark features of the illness. The first that shows up are of these rather strange intraoral lesions called Coplic spots. Those are probably, along with a fairly typical fever and neuralgia and general sense of lethargy, that the Coplic spots are one of the very first indications that the reason for the sickness is attributable to measles virus. The rash that is where measles got its name, measles actually, the word itself actually means many little spots, as it turns out. The rash itself doesn't occur until 7 to 10 days following the initial exposure. So oftentimes individuals will be walking around feeling sick, feeling flu-like in a sense, will be acutely infected with measles virus, obviously at risk for spreading the infection then to other individuals that they encounter who may be otherwise unprotected. And the true diagnosis for measles virus wouldn't actually become apparent until the rash is evident. And that usually doesn't happen until much later in the infection when the virus is able to spread to the skin.
So when I was a kid, we didn't have measles vaccine and was part of the childhood infectious diseases that one got. Everyone got the rash. Of course, there were a couple of different rash diseases. We got chickenpox also. Some of us got mumps.
Right, rubella would be one.
Rubella.
Right.
And so is measles sufficiently different in the rash that you can tell the difference, or is it not?
Well, it was initially when it was first identified, it was distinguished from smallpox actually by the type of rash that occurs. I think what's notable still about the measles rash is that it virtually covers the entire body. So you get in the rash are these very small. They tend not to be purulent, so they tend not to burst. You end up getting the rash all across the body, whereas for some of those other infections that you talked about, it's more focalized on the body.
So if you have a period of incubation during which time you have maybe nonspecific viral symptoms, unless you're very alert and you see the Coplic spots, and during that time you're shedding virus, I presume, right?
That's right.
So that is how other people get infected, because you have someone going to school and it's not clear they have measles, and then they spread virus, and then when they get the rash, they stay home. By then, it's too late.
Right. And this is one, even among other respiratory viruses, measles virus is considered to be just extraordinarily contagious. So there was an incident even fairly recently of a woman who returned from Europe and was at a party with a couple hundred individuals. So this was all taking place in a very large venue, and virtually all of the uninfected individuals in that room subsequently came down with measles virus infection, just as a consequence of being in the same fairly large environment with this infected individual. So it is a remarkably robust pathogen in terms of its capacity to spread within a population.
Do we understand why that's so?
No, actually, we don't, in fact. I mean, obviously this virus, like any respiratory virus, is transmitted by aerosol. So if this virus makes you cough or sneeze, you generate a fair amount of liquid volume that can, depending upon droplet size, can be, carried in the air for some distance. But why uniquely measles virus is so transmissible relative to other infectious or respiratory pathogens isn't entirely clear.
So you inhale the virus basically, and it goes into your respiratory tract where it begins replication.
That's right.
And then what happens next?
So the illnesses that occur are coincident with where the virus is replicating. So the very, as you point out, the very first place that the virus replicates is in the respiratory epithelia, and this ends up resulting in the congestion, the fever, the kind of common cold symptoms that is sort of the first stages of measles infection. And then eventually the virus actually is able to translocate or move to draining lymph nodes and can infect lymphocytes. And actually most of the mortality that we talked about before isn't due to the measles infection directly, but rather due to the immunosuppression that measles virus causes. So I suspect most of your listeners are aware that HIV causes immunosuppression, and it does so by wiping out CD4 positive T cells in the human. Well, measles virus is another virus that can cause immunosuppression. Unlike HIV, that immunosuppression is a transient one. So you are immunosuppressed, in a sense, just during the period of time that you're harboring measles virus. But in parts of the world where, let's say, sanitation is not optimal, that period of immunosuppression may be sufficient for you to become infected with other pathogens that may then be the basis of the subsequent serious disease and, in some cases, death.
So when people die of measles, is it typically because of a secondary infection of some kind?
Right.
Yeah.
Right. Most often. So, I mean, there certainly are direct consequences of measles infection itself, right? I mean, so you can end up having pneumonia, for example, or encephalitis, which we may talk about in a bit. But in fact, oftentimes gastrointestinal illnesses, diarrhea, may be the principal reason by which measles virus causes mortality in an indirect way. That is, you're immunosuppressed and therefore you're developing illness as a consequence of all the things that you get during that period of immunosuppression.
So with HIV, there are very typical secondary infections that you get. Are there such typical ones for measles?
Well, I don't think that there are specific ones that are characteristic for measles virus infection. I think many bacterial infections, parasitic infections, as I say, based upon the environment in which the infected individual is and what kinds of pathogens are endemic within that region, would probably dictate what the most likely consequence of the infection would result in.
So in the U.S., secondary infections are not as common, basically.
No, that's right. And that's why mortality as a consequence of measles virus is kind of an unmeaningful definition, because in the United States, fortunately, measles viruses, while it still can result in serious consequences like encephalitis or pneumonia, is relatively well controlled because you can put a kid on antibiotics, for example, if they come into the hospital. But in developing countries where medical care is perhaps not as ubiquitous, that poses much more of a challenge.
Now, you may have mentioned this, but what actually causes the rash?
Well, you end up getting, after the virus replicates in the draining lymph nodes and causes the immunosuppression, it eventually goes to, it can infect the epithelium, the skin. And when it does so, it causes, measles virus induces fusion of infected cells. And so these rashes that you end up getting are a consequence of I believe, are a consequence of fusion of infected cells that are present on the skin.
With measles vaccine, you get 1 or 2 or 3 doses when you're a child, and that's it for the rest of your life. You get lifelong protection. Why is that,
That's right.
and why is that different from influenza?
Right. So, well, measles virus is, as you mentioned before, it's a member of the paramyxovirus family. It's a negative strand RNA virus. So, in that sense, like influenza is an RNA virus, or like mumps or rubella are RNA viruses. Most RNA viruses tend to have a fairly high mutation frequency. So the reason, of course, why you have to be vaccinated each year with influenza is because the virus is constantly adapting. And then in some cases, as we're seeing this year, you end up having major changes where you have a whole reassortment and rearrangements. So you end up generating an entirely new virus. For measles virus, actually, it is not particularly mutagenic. It doesn't have a very high mutation frequency. And so therefore, by being vaccinated with the live attenuated strain, and initially it was just given once, and it was found that a small number of people, maybe 5% of individuals or so, didn't have lifelong protection. And that was the basis for the 2 vaccination schedule that continues today. But that 2 vaccination schedule is sufficient to protect people for a lifetime. Mm-hmm. There were actually really fascinating studies that were done of individuals on the Faroe Islands, which are this little group of islands in between Europe and the United States. And when travel was initially occurring between the 2 continents, the Faroe Islands were used as a stopping point. And when sailors first arrived on these islands, they brought with them all the diseases from Europe that they carried, including measles virus. And a high proportion of the indigenous Faroe Island residents became infected, as you would have predicted. And then the shipping routes apparently changed, and the Faroe Islands were no longer a stopping point along the way across the ocean. And so for approximately 70 years or so, there were no cases of measles virus. And subsequently, when those shipping routes changed yet again and the Faroe Islands were once again a stopping point, measles was reintroduced. And in fact, all of the individuals that had been infected when they were children and now were much older individuals in their 70s or 80s remained protected once again. And those individuals that had not been previously infected were now susceptible to the infection. So this was I think, a really interesting natural protection experiment that showed that memory to viruses can last for, in fact, an entire lifetime, for decades, even in the absence of constant restimulation by the infection.
Yeah, I think those findings were important in our developing modern vaccines. It was an important
That's exactly right.
That. So how far back can we, you mentioned the Faroe Islands, this was in the 1800s?
Yes, that's right.
So how far back can we see measles in history?
Well, so it's presumed, or the measles virus is in a family of viruses with 2 other pathogens, but those are not human pathogens. So rinderpest virus is a virus principally of cattle and of goats and of other, I believe it's called cloven-footed animals. And then the other one, which is in fact a serious veterinary pathogen, is called canine distemper virus. So these 3 pathogens all are part of the morbillivirus family within the paramyxoviruses. And it's believed that when back probably about 5,000 years ago, when cattle were being domesticated and used used by farmers, that there was some transmission of the original rinderpest strain that became adapted and infectious for humans. So that probably took place, 5 millennia ago or so.
So measles is a zoonosis, basically.
It's exactly, yeah. That's the belief. That's right. And that all sort of originated probably in the Middle East, and then was eventually brought to the Americas by colonization and migration, and it ended up causing lethal effects on Native Americans as well.
Yeah. There's a lot of good history about that, actually, how it was brought around the world by explorers, and
that's right.
Native peoples didn't have the virus, and they were wiped out in many cases.
Indeed.
Yeah. Now, you talked about how we get a rash, we get fever, typical virus symptoms. You get, and you mentioned encephalitis as a possibility. So what happens there? Right.
So one of the more interesting characteristics, and actually one of the certainly one of the more frightening characteristics of measles virus is its rare but very, very serious potential to infect the human central nervous system. So for most individuals, you get infected, you make an immune response, you do go through this period of immunosuppression, which we can talk a bit about, but most individuals end up recovering from the infection and then are afforded lifelong immunity. But for an extraordinarily small proportion of cases, the virus is able to make its way into the central nervous system. And when it does so, it can result in a number of central nervous system CNS diseases with impossibly long names like subacute sclerosing panencephalitis or measles inclusion body encephalomyelitis. They're different. These CNS diseases are somewhat different from each other in terms of the kinds of individuals that get infected. But what makes them rather remarkable is that you don't actually end up developing signs of those CNS diseases for months and often years, perhaps even a decade after the acute infection. So, for example, if a child is infected acutely with measles virus at the age of 2, you may not actually begin to see signs of SSPE, one of these CNS diseases, until that child is 10 or 11 years of age. And it initially manifests as minor cognitive changes. The child may not be paying as much attention in school or may have other what appear to be fairly subtle cognitive changes. And then it becomes more sort of fundamental changes where you end up having seizures and paralysis that eventually leads to coma and more often than not ends up leading to death. So while these are rare infections within the central nervous system, they're almost invariably fatal. And because there are no post-exposure cures for measles virus, there's really very little that can be done for these patients. There's oftentimes interferon is given in very high doses, and that has some efficacy, but it still is not a terribly effective way to rid the pathogen.
But do we have any understanding of how this virus, this is what we consider an acute virus infection, how
Right.
it persists for years in someone?
Right. So there's a couple of, I think, really interesting points to make on this. So one of them is that there are a number of studies. These are basically PCR-based analyses where tissues have been taken from individuals who died of non-viral causes. Actually, these are principally Japanese-based studies. So individuals who died, let's say, in their 80s, where tissues were harvested from these individuals and where PCR was done to look for the presence of various pathogens, including measles virus. And what was really interesting was that in a very high proportion, let's say maybe 25% of these individuals who died of, let's say, heart attack or stroke, something that we presume wouldn't be virus-based, they were able to find evidence of measles virus, and in particular within the central nervous system of these individuals. So the point that if these studies are in fact the case, then what this would argue is that measles virus may actually be more of a persisting pathogen than we otherwise would have imagined, but for the vast majority of individuals, not necessarily have a pathogenic consequence. So it may not simply be that the important factor is, does the virus gain access to the central nervous system, because that may actually be a more common event than we would have suspected, but rather what happens to trigger the virus to replicate in a robust way to result in these lethal CNS diseases like SSPE. In part because they're rare, we don't know whether during that, let's say, 10-year period, that latency period where the child is fine, we don't know if the virus is replicating at an incredibly slow rate and then, inexorably reaches some percent of neurons or percent of cells that are infected that then trigger the overt symptoms, or whether the virus is completely quiescent during that period and some trigger causes it to reactivate, similar to what happens for the herpes viruses, for example. We have no idea about that because the number of human cases is small, and because until fairly recently there have been no good animal model systems to study measles virus.
So we can assume, though, that the viral genome is present in these children for all those years, or is that even open to question?
Right. So the basis, no, it's fairly clear that the genome is present in these kids. I mean, the reason why it's known that measles virus is the trigger of these diseases that occur so much further down the road is, in fact, the presence of measles virus within brain tissues that are isolated post-autopsy.
So what kind of animal model do we have now that we can use to study these issues?
Right. So, and here we took a page actually from the Racaniello book, in the sense that for a lot of infections, a lot of viruses of substantial impact on humans, whether that's HIV or poliovirus or measles virus or the hepatitis viruses, one of the real challenges, if you want to use mouse models to better understand the pathogenesis of those infections, is that mice are not permissive for those pathogens. And so you were the first to show that if you identified the human receptor that allows the virus to gain access in a human, and now you genetically engineered animals, mice, to express that receptor, you stood a chance of overcoming the barrier to infection, so that now what might normally not be able to infect mice may now be able to use that transgenic receptor as a gate to gain access to mouse cells and initiate replication and cause disease. So in 1994, one of the first human measles receptors was identified. It was a protein called CD46. And with the identification of that receptor, it became possible to make transgenic animals which expressed CD46 in the hopes that we would be able to generate transgenic animals which were permissive for measles virus infection. And that was successful to a point. So we can talk about this more later, but one of the mouse models that I work a lot with along with people in my group, is an animal model in which we expressed CD46 specifically in neurons of mice. And while those animals have been helpful for us in understanding how viruses spread within neurons and how immune responses are generated within the central nervous system, we want to be very careful not to draw too many comparisons to what happened in people. In humans, the virus obviously first replicates in peripheral tissues like the lungs and the blood, as we talked about, and the skin, and then somehow is able to make its way up into the central nervous system in a rare number of cases. This mouse model is quite different in the sense that the only cells that are permissive, the only cells that express this receptor, are neurons within the CNS. Other models have been made in which CD46 or subsequently another receptor has been identified. So we're up to 2, and everyone believes there are many more still to be identified. But other models mice have been made in which SLAM, SLAM stands for signaling lymphocyte activation molecule, have been expressed in transgenic animals to mimic some of the immunosuppression features of measles. And obviously that has direct relevance to the human biology because, as we talked about before, I would say 99-point-something% of the reason why individuals die as a consequence of measles virus is due to immunosuppression. So that's really going to be a critical feature for us to develop a tractable mouse model system that mimics the immunosuppression we see in people.
Yeah, it's typical. That's why we call them animal models, right?
That's exactly right.
They never completely mimic. Even for polio, the transgenic mice are not an exact mimic because you can't feed them virus and get them to be infected unless you do unusual things. But you can learn a lot. So why do we think there are more than 2 receptors? What's the evidence for that?
Right. So this has been a huge an interesting controversy to be part of in the field. When CD46 was first identified in 1994, it was identified principally using the strains that most measles virologists used, which in fact were vaccine strains. So Edenston, for example, is the classic measles virus strain that most of us end up working with in our laboratories. But there was always that undercurrent, that sense that possibly the receptor that the vaccine strains utilize may be different from the one that wild-type strains use. And indeed, in 2000, I believe it was, a lab identified SLAM as being a wild-type receptor for measles virus. And subsequent studies actually showed that CD46 indeed was a receptor for measles, but principally the vaccine strains of measles virus, whereas SLAM seemed to be more relevant to, or more, in a sense, the receptor or permissive for wild-type strains of virus. The problem is, though, that SLAM is restricted. As I said, it stands for signaling lymphocyte activation molecule. So its expression in humans is very restricted. It's expressed on some T cells and on B cells. And as we talked about before, one of the characteristics of measles virus is its capacity to infect many, many different tissues in humans. So there still is the belief that there is another wild-type receptor as yet to be identified out there that would allow for permissivity or infection of non-lymphocyte cell types.
So for polio vaccine, we know exactly how many mutations distinguish the vaccine from the wild-type virus. Is that also the case for the measles vaccine?
Right. There's one particular residue actually within the hemagglutinin that seems to be quite important. It's a residue that probably very modestly changes the conformation of the hemagglutinin molecule that dictates whether it has preference for CD46 or for SLAM. And indeed, this is the residue. I believe it's a residue 481 on the hemagglutinin molecule of measles virus. That modest little change influences which of the receptors is utilized. I mean, just to be clear, it's not black and white. It's not as if only the vaccine strains utilize CD46 and only wild-type strains utilize SLAM. There's a little bit of crossover, but there clearly is a preference, and that preference seems to be influenced by a single residue.
That's amazing. And and this, of course, was a vaccine that was derived empirically in the 60s, right?
That's right. That's right.
So this is injected. This vaccine is injected, correct?
That's right. Yes, along with it's along with the mumps and the rubella vaccine. So it's part of the MMR vaccine, and in fact, there are some companies that want to now add a fourth to that cocktail, the varicella, which is for chickenpox.
And these are all infectious virus vaccines, so they all multiply. And in the case of measles, as you said earlier, the virus multiplies. Do you get any of the symptoms of measles as a consequence of vaccination?
Well, that's actually an interesting question. I mean, I think it is fairly typical for kids to have a day or two of low-grade fever following a vaccination visit to their physician. Oftentimes, kids will get multiple vaccines. So not only will they get measles, mumps, rubella, but they may also get DPT. They may also get poliovirus vaccine. So they may be given actually quite a variety of different vaccines all in the same visit. And of course, the intention of these vaccines, whether they're live attenuated or whether they're subunit vaccines, the intention is to induce an aggressive immune response that's going to be protective.
Right.
But making an immune response alone can actually make you feel fairly poorly. You can produce lots and lots of cytokines that are going to give you a low-grade fever and make you feel somewhat sick. So I think that's not unusual at all for kids to have a day or two of feeling not optimal subsequent to vaccines. But then, of course, the intent is that once that goes away, you're left a protected individual from those pathogens that would have a much worse consequence on you should you have been infected in the first place.
So you mentioned earlier that one of the problems in effective vaccination is maintaining the cold chain, and that could account for some of the cases of measles that we still see. But what else is a problem with measles immunization?
Right. So this is, I think, what people, at least in the United States and in Europe, know most about measles virus is that there's been a very, very public and contentious and now actually long debate about what role vaccines, including the MMR vaccine, may or may not play in childhood autism. And as a consequence of some publications that came out in the late 1990s, there has been a very public discussion between physicians and parents about vaccine safety, and there have been a fair number of parents who've elected to not vaccinate their children for fear that vaccines may to some degree contribute to autism in kids. And this, of course, then leaves children unprotected and decreases the whole basis of herd immunity. So the idea that the more individuals within a given population that are infected, the more, in a sense, strong the wall or fortified the wall is around that community to block out the ability of pathogens to gain access. So as that rate of vaccination decreases, the chance that these viruses, measles, mumps, rubella, others, can gain access to that community increases. And it's not simply to put the children who are unvaccinated at risk. Viruses, of course, can mutate and can develop, so that if a mutation were to occur in this population, that potentially could then spread to children who've been vaccinated but may not be, in fact, immunized against that new variant of the virus. So preserving herd immunity is a key theme or a key component of vaccination. And to some degree, communities which have elected to reduce their rates of vaccination or to delay when children are vaccinated have in fact had a higher rate of infections.
So as you know, the World Health Organization wants to target measles for eradication once they're done with polio. And in light of this issue, the parental anti-vaccine stance, how are they going to be able to do that?
Yeah, so I'm not sure I have an answer to that. I do think that the challenge to the WHO has become more acute as a consequence of this discussion. I mean, so, coming back a little bit to autism, and the reason for why this theory or this link was initially proposed is that autism, which is a CNS disorder that becomes manifest usually somewhere around the age of 2 in children. It's defined principally by attenuated social interactions between autistic children and the individuals around them. The rate of autism has increased dramatically in the US and in Europe. So we're on the order of 1 in 150 kids now are diagnosed with some component of autism spectrum disorders. And that's actually important, the name, because it's not a single kind of disorder. It's actually a whole family of very, very diverse disorders. So Asperger's syndrome, classical autism, and then the third component of it is called PDD-NOS, for pervasive developmental delay not otherwise specified. So it's kind of a catch-all for a variety of other neurodevelopmental disorders. But the rate of the frequency of these disorders has increased really, really dramatically over the last 10 years. And there are some that believe that it's just that we've gotten better at diagnosing them, but there are, I suspect, many more who believe that there is something that has caused autism to go into almost epidemic proportions. And so people have therefore naturally looked to see, well, what's different about our environment that might dictate why our rates of autism in 2009 are so much more than they were in 1989, for example. And there have been some that have made that connection to increasing numbers of vaccines that children end up getting, or the kinds of preservatives that are included in some of the killed vaccines.
So is there any evidence that supports that hypothesis?
Well, the initial papers that came out, the one paper that came out in 1998 that really sparked this issue for the MMR vaccine looked in gut biopsies of children with and without autism spectrum disorder. And the conclusion from the paper was that the children that did have autism had measles virus RNA present within those gut biopsies, and that the children without did not. And the paper was actually, in that case, fair, and it didn't try and say that vaccination causes a chronic infection in the gut that somehow leads to the etiology of autism. It didn't actually say that. But a lot of people, including the lead author, then subsequently, in discussing the consequences of this, in fact did say that there was the perception or the belief that vaccination may be in some sense linked to autism. That's really been the only report that suggests a link between vaccination and chronic infection and autism. There have been actually many studies prospective and retrospective epidemiological studies that have followed up on that observation to look and see, well, in fact, if we look at kids who have been vaccinated versus those that haven't, is there an increased risk of autism? Because that would be an obvious prediction from this. And there aren't. So of all of those other population-based studies, there is no connection, no evidence for a link at all between vaccination, either with MMR or with any other combination of vaccines, and autism. That said, there are many parents who very strongly believe that their child began to show regression signs of autism subsequent to vaccination, so that their child had communication skills, made eye contact, played interactively with other kids, then got vaccinated, and then began to regress. And these parents are understandably very passionate about this feeling, because even though it's not science the way we think of it, it is for them, their child, and what they've observed. And there really haven't been any studies that have looked specifically at those children who have regressed closely linked to vaccination to see if by some chance there is something that perhaps is genetically unique about those kids that might in fact have caused vaccines to trigger, let's say, an altered immunity that may in some sense have been connected to autism. We know so little about what causes autism in the first place that there are still a fair number of open-ended questions that have yet to be addressed. So, but to answer your question, no, there really is not, other than that first paper, sound scientific evidence that suggests that there's any connection at all. And this is the reason why pediatric societies are so adamant about saying, please vaccinate your kids, because it's more important that you're protecting them against these very serious human pathogens than the potential risk that might be associated with autism.
And the problem, of course, then extends to other vaccines as well,
Right.
have not been implicated in any study yet. They're a vaccine, so by extension, the parents who are suspicious of MMR are suspicious of all of them. And that's tragic, right, because all these diseases are preventable.
Right. So I mean, to back to your, to the question about the World Health Organization, the challenge, let's say maybe 20, 25 years ago in eradicating pathogens like measles virus was simply getting the vaccine into the individuals that were, that needed to be vaccinated. So it was simply a practical issue of how do we keep it cold and how do we make certain that we, inoculate these kids at the right time postnatally so that they're protected, so that we can wipe out measles virus. But now it's become a much more challenging problem, because now you don't just have to inject the patient, you need to convince the patient, right? And you need to compel individuals that the vaccination is something that's going to be important to do. And I suspect that strategy is going to be fundamentally different from what the World Health Organization initially, crafted as their plan for eradication of measles virus worldwide.
Yeah. When they crafted it, I don't think this whole issue was.
No, not at all.
So do you think that measles is eradicable?
I suspect it is. I mean, it has elements of, so humans, there's no, at least that we're aware of, there's no animal reservoir for it. So it's not as if it's, lurking out, where we can't gain access to it or we can't vaccinate it. So, I mean, it has elements of a eradicable kind of a pathogen. But, bear in mind what I'd said at the outset, that still, as of, this past year, millions, and maybe on the order of tens of millions of individuals are still acutely infected with this annually. So we are still far away from being able to fully eradicate it. So I guess I'm going to sort of fence it in my answer and say, yes, I do think it is eradicable, but it's not going to be something that I think happens in a very short time frame.
Technically, polio is eradicable also.
Right.
And we're almost 10 years behind schedule there. So,
right.
I mean, many other issues come into play besides the biology of the virus when you talk about eradication. And that's what's happened with polio and will likely happen with measles as well.
Right. I mean, I think, distinctly different from smallpox, where your chances of dying as a consequence of smallpox were 30%. As serious as measles virus is, your chances of dying from measles virus are far lower, even in developing parts of the world. So perhaps the issue is not necessarily eradication of the infection, while that is certainly a goal for the future down the road. Possibly a shorter term goal is to make certain that patients that do become infected with measles virus are well taken care of. So ample antibiotics, for example, to cover them during the period of immunosuppression, for example.
So Glenn, that was a pretty good discussion. Anything else we ought to cover before we move on to email?
The only other thing, and it may be a little too, I'm not entirely sure whether your audience is going to be interested in this, but the basis for how measles virus causes this transient immunosuppression.
Oh, yeah.
We could talk about a little bit.
I think that's worth exploring, yeah.
One of the things that's an active field of study for measles virus biologists is understanding how this virus is able to cause immunosuppression. And I suspect that all of us in the field would concur that that's the key question to address. And what's really interesting, at least to me, is that there are many theories that have currently been proposed, and probably to some degree all of them are right in terms of how this virus is able to induce this transient immunosuppression. I mean, it's a bit of a mystery because during the period of time that you're acutely infected, you're globally immunosuppressed. That means that your whole immune response is attenuated or weakened, and therefore you're permissive to infection with many different things. But if you look at what proportion of your immune cells are actually infected with measles virus, it's a very small percent, on the order of maybe 1 or 2% of your T cells are infected. So how can infection of such a small proportion of your immune response have such a global kind of impact? And there are many, many different theories. There are excellent studies that have shown that measles virus can block proliferation of immune cells, that it can disrupt maturation of immune cells. But there's one study that has actually been shown in vitro and then correlated or shown to also be the case in acutely infected individuals that I think is fascinating. That when you make an immune response, you have a particular kind of immune cell called T helper cells. And those T helper cells can produce two different profiles of cytokines, and they're called Th1 or Th2. The Th1-based cytokines, which include ones like interferon gamma, are particularly useful for getting rid of cell-associated pathogens, like viruses, for example. The Th2 cytokines, like IL-4 and interleukin-5 and interleukin-10, are particularly helpful for getting rid of extracellular pathogens, ones that don't require replicating inside of a cell, like bacteria and parasites. And how your immune response knows whether to make a principally Th1 or a Th2 profile is actually dictated by another cytokine called interleukin-12. And interleukin-12 is a really key player here because it's made by antigen-presenting cells, the very first cells that gobble up the offending pathogen. And interleukin-12 production skews an immune response toward a Th1 profile. So that if you make interleukin-12, you're going to principally make those same cytokines that are going to be useful for getting rid of a virus. And what Diane Griffin and her colleagues had shown some years ago was that measles virus infection of antigen-presenting cells blocks IL-12 production. So in fact, it's not that you're immunosuppressed as much as it is that you're making the wrong immune response. In the absence of interleukin-12, you're not making a Th1 response, you're making more of a Th2 response, a response that would be beneficial and useful for getting rid of bacteria and parasites, not getting rid of the measles infection. And this has later been shown to be the case in acutely infected individuals where they have a high level of these Th2-like cytokines that are in the circulation. So it's a very sort of subtle way, but obviously impactful, a very subtle way by which measles virus is able to, in a sense, fool the host response so that it's not that it blocks its capacity to make a response, but that it's making the wrong response, therefore enabling the virus more time to replicate and, in a sense, do its job.
Yeah, and the side effect is that other pathogens can get in.
That's right.
So why doesn't the measles vaccine have this effect on IL-12 production?
Well, it actually may. I mean, even in acutely infected individuals, you end up having, eventually you make an immune response that is protective. So it's not that it is that you're unable to make the right immune response. It's just that response is delayed or is less robust at the earliest stages post-infection. I don't believe that anyone has actually looked to see whether this IL-12 suppression is unique to wild-type strains or whether it also occurs for the attenuated vaccine-like strains. I don't know if that's been done.
Is it worth thinking about IL-12 therapy, replacement therapy for people who are immunosuppressed after measles infection, or are there other ways to deal with that?
Well, that's an interesting theory. I mean, of course, as we were saying before, cytokine production has its own downsides, of course, right? So production of IL-2, for example, causes is the basis for much of the reason why you feel sick following infection. So how IL-12 administration to an acutely infected individual, what kinds of medical consequences that would have, and whether you need to deliver it in a very particular specific way, that is where T cells are being educated within the lymph node as opposed to just, infusing it into the blood and, in a sense, perfusing the individual with IL-12, it is not entirely clear.
Well, that's great. What I would like to do is have you back, and then you can talk about your own research, because I know we didn't get to do that, but our listeners would love to hear it. And it sounds like you enjoy TWiV, so we'll have you back.
I do. I'm an avid listener.
Excellent. Well, let's do some email, and
Sure.
You can hear what kinds of wonderful questions our readers send in. The first one's from Natalie, who wrote, "Dear TWiV, thank you for your amazing podcast. Your program is a great way to have fun and learn. I am a graduate student in microbiology at the University of Iowa. I will graduate in 12 to 18 months and am thinking about what I want to study in my postdoc. I would like to know what each of you would pick as key areas or questions in virology that need answering. In other words, what do you think the seminal discoveries of 2010, 2011, 2030 will be? Thank you again for fun and facts."
Wow. You get to go first.
You want me to go first? As a guest, I was going to let you take the first.
Oh, that's very kind. I'll be happy to take a crack at it, actually. I think one thing that's becoming clear is that, and I have to be careful because, of course, you've written one of the definitive textbooks for virology, but one thing that's becoming clear is that viruses don't have one replication cycle, one life cycle. When we study a viral life cycle, we're looking at it in one particular cell population or in one particular cell type. And I think what's becoming clearer, at least in the literature that I read, is that that is strongly influenced by the cell itself. So the cell is not just this inert bag, but it actually provides, obviously, many, many proteins that are necessary for the virus to complete its life cycle. So if that's the case, then different cells are going to provide different toolkits, in essence, for these viruses that may actually strongly stratify the kinds of infections that you end up getting. So one of the things, for example, that my lab looks at is viral infection in neurons. And we know that measles virus infection in neurons is fundamentally different from measles virus infection in a fibroblast or another non-neuronal cell population. So I think as time moves forward in virology, we're going to begin to appreciate these cell-specific differences and begin to understand what the cellular tools are that viruses use. And it's going to be not only helpful in understanding more about virology, but in using viruses, in a sense, as mini flashlights to understand more about cell biology as well.
Well, my ideas are pretty broad, and one of them is viral discovery. I think that we need to know what viruses are out there. And it's not just a matter of finding what virus is causing a new hemorrhagic fever, but what viruses are in humans at a steady state level or in animal populations. And this is really, we talk about this all the time on TWiV.
Right.
There are a couple of labs that do that, the Lipkin lab, the DeRisi lab, and there are a few others, but there aren't enough, and those labs are oversubscribed. I would do a postdoc and learn the techniques of discovery and go out and look at viruses in rodents or in bats and just see what's out there, or even in people. We have no idea. Someone asked me, someone sent an email to say, what we know about the bacteria on the skin now, what about viruses? We really don't know.
Right.
How we're colonized. I think that's going to be important.
Right.
Pathogenesis, as you said, is huge. Now we can do so many experiments, and particularly the immune response and how viruses overcome the immune response. I think the only reason we have infections is because this wonderful immune system that we have gets compromised by viruses. And how do they do that? We know lots of ways, but there are doubtless many more, and that will help you treat diseases, which is my other area, which is antivirals. I think we have far too few antivirals, and we need more, and we need people that know how to do virology in order to look for them. So those are sort of three. Pathogenesis is very broad, but I think it's where virology is going.
Yeah,
well, it's already there. In fact, the old days of studying replication are almost over. I'm on a study section, and I know that a lot of grants are all dealing with pathogenesis now, and trying to understand what a virus does in cell culture is still important, but when money's limiting, the money is going to pathogenesis.
Right. Well, I mean, I don't know about what it was that got you interested in viruses in the first place, but for me, it was absolutely the fact that these have clinical impact, that these are pathogens of humans, that they're fundamentally different from how bacteria replicate, how parasites replicate, and just understanding the unique interaction between a virus and its host was what got me in the field in the first place.
Absolutely. And I said many, many months ago on TWiV, I read Fever in the 1960s, the discovery of Lassa, and that got me interested in viruses. It's the same thing.
One last quick point is that viruses are actually now being utilized as vectors as therapeutics. So we shouldn't just necessarily look at them as, evil things to eradicate or to prevent. That there's the potential actually also to use viruses as therapeutic tools.
Absolutely.
So Measles virus, in fact, which we've just been talking about, there are a number of labs that are looking at using measles virus as an oncolytic or tumor targeting pathogen to eradicate tumors in individuals, because it turns out that some tumors express more of the measles virus receptors than the surrounding non-tumor tissue. So viruses may not necessarily, in future work with them, may not necessarily be just about eradicating them, but using them for good.
Yep, absolutely. Yeah, we in fact have a couple of requests to do a TWiV on using viruses as anti-tumors. There are plenty of examples of that, and we will do that one day. Our next email is from Peter, who wrote, "Great show again, episode 36. So this is 2 episodes ago. I meant to read this last week. I'm sorry, Peter. And we had talked about using MDCK cells to grow a flu vaccine, and I had expressed skepticism that they could be used. Well, it turns out that, in fact, Hamish was right. Novartis uses MDCK cells, and this was approved in June 2007, and he sent a link for that. And he also has links for some other different cells used to grow various vaccines. Baxter uses Vero cells, and then he notes, "I'm wondering what had happened to Sanofi Pasteur's Par-C6 cell-based vaccine. The story is almost 3 years old, but they were writing about a successful 20,000-liter bioreactor run, which is as big as it gets, I suppose."
Imagine so.
20,000 liters.
20,000.
I was also wondering if Vivalis's EB66 cell line might be used for influenza vaccine production. The companies who licensed it includes the usual suspects, I mean the largest flu vaccine manufacturers, and we have a link for that as well. Last but not least, there is Novavax using insect cell culture and recombinant baculovirus in combination with disposable bioreactors. And, of course, that fits in with Protein Science, a different company that we talked about today, using insect cell culture for flu HA-based vaccines. And he writes, "Although currently I am doing large-scale cell culture for manufacturing of therapeutic proteins, monoclonal antibodies, and fusion proteins for a living, vaccines, especially viral vaccines, always interest me. Speaking of immunity, there was a blog article by Mark Crislip on the Science-Based Medicine blog recently on herd immunity," and he sends a link. "Could you elaborate in a future episode on the topic? Why are there diseases which could be eradicated, smallpox, while other are possible to be eradicated, polio, and others are not?" And absolutely, that's a great idea for a topic, so we'll do that. Thank you very much for TWiV. Your podcast makes me look forward to Monday, which means a lot, I guess, when it comes out when it comes out in Korea, where I live. Thanks for that, Peter. So this shows you the breadth of people that listen to TWiV. They're doing all kinds of things, and we get great questions and comments. I'm very proud of all our listeners.
Right. Well, this is why I said to you before why I think this is such a great idea, because there are all of these fascinating discoveries and observations that are made, and part of our responsibility, I think, as scientists, is to come up with ways to communicate this. I mean, oftentimes, at least in the U.S., this work is being done on on the taxpayer's dime, in a sense. So as a way to come up with novel strategies to communicate not just the details, but also the enthusiasm, the passion for the work that we do. This strategy, this approach is terrific.
Yeah, I agree. You're preaching to the converted, obviously.
I suspect.
But it's always good to emphasize. Ricardo wrote, I really enjoyed TWiV 35. I'm reading a previous recommendation of TWiV, Microbe Hunters, and it is all very exciting. Here is a great software for history telling. It allows a timeline presentation as well as keynote integration. And he sends a link to a program called Timelines 3D, which we'll provide in the show notes. Just one question. I'm preparing a practical laboratory virology class. I'm looking for a source of lab protocols for undergrad students. Any suggestions? A book, website, anything? I've found a lot of bacteriology, but not much luck on virology. And Ricardo is a professor in Portugal. Well, I think you should go look at a site called All the Virology on the World Wide Web, www.virology.net, which will, in fact, be my pick of the week because we've never mentioned it on TWiV. It's a great compendium about all things, and there are plenty of links to educational virology tools on that site. You know anything else aside from that, Glenn?
No, actually, when you were reading the question, that's exactly what popped to mind. I mean, also, most investigators are quite willing to share their protocols, so you may not have sufficient detail in the materials and methods of a paper, but if you use that as the springboard for contacting them directly, many PIs will be quite willing to send along their more detailed protocols as well.
Absolutely,
so,
yeah. In fact, so check that out, Ricardo. And if you don't find anything you like, let me know, because Hamish Young used to do virology labs with the medical students here, and he might have the protocols from that. And our last one, I'm going to play our first audio email file from Jim. And Jim took up my challenge to send in feedback by MP3 file, and so here it is.
Hello, this is Jim Vandiver, retired power plant worker from rural Virginia. I've listened to TWiV number 35 twice with the same delight as the other 34 episodes. While mulling over all the interesting information in the last one, it suddenly occurred to me that the dynamic complexity of your sessions are much like the viral world. So yours must be a viral program, I think. Now, the mention of rat feces and urine as viral sources in podcast 35 are linked, to my mind, to such things as the face masks discussed at other times that raise other questions, and that leads me to ask you guys to consider having a topic I call design considerations. Or, what does a virologist think of this? For example, here's a list of items. Will replacing vacuums with mops, or using wet vacs only that use bleach solutions, reduce the spread of viruses? Or should brooms be banished because of the way they stir up dust? Also, should central ventilation systems only be used in private homes and then only have outlets high up on the wall and returns at floor level, with a mandatory UV light placed at the cooling coil? Should vehicle ventilation systems also be equipped with UV lamps? During viral outbreaks, should pets be periodically wiped with a bleach moistened wipe, as well as other highly handled or highly touched surfaces in the home? Should toilets be equipped with lids that fit more tightly and have to be closed before a toilet can be flushed? Should men sit to pee? During a pandemic, should banks apply disinfectant processes to all money passing through them? Should postal facilities do the same for mail? Would silver-plated handles on doors to public places and restroom fixtures reduce transmission of viruses? Oh, I forgot who mentioned the fascinating fact in session 35 that no domesticated animals come from Africa, but it occurred to me that the origin of man is supposed to be in Africa. If that's so, does that mean civilization is doomed because man can't be domesticated? Then Alan recommended Zotero as a Firefox extension. Another extension that I use is called Juice, J-U-I-C-E. It allows searches from within a web page. You just highlight a word or phrase, then drag it a short distance to activate the program. It then opens a sidebar and displays the search results. It's an easy and convenient way to answer questions about items encountered while reading material within the browser without leaving the page being viewed. And lastly, this audio file was speeded up 150% using software called MP3 Speed Changer. That comes from a website called crazyboomerang.com. That's CrazyBoomerang, all run together. It took me about 6 hours to construct this presentation, write it out, record, and send it in to TWiV. So it's a tedious process for some of us, I think. Thanks for listening, guys. Bye.
Some interesting ideas there. I think you always have a trade-off in life between the things you do to prevent infection and how prevalent infection really is. So animal feces and urine is not a problem in a lot of places. And so you wouldn't have to go through these procedures. You also have to consider that bleach is pretty caustic.
But it brings up, actually, to me, it brings up this interesting interface between virology and economics in some way, right? I mean, that a lot of things that we potentially could do that would limit the spread of pathogens, viruses, or otherwise, might actually be financially so burdensome that the benefit that you gain from reduced pathogenesis of these pathogens would not be as great as the cost to society would be.
Sure, exactly.
And how you make those decisions, how you take best advantage of trying to protect public well-being while at the same time recognizing that finances, money isn't unlimited, is I think is a kind of a fascinating topic. It's very pertinent to vaccine development, for example.
Sure. Now, I know the perfect person to do a TWiV on this topic. His name is Harley Rothbart. He has a blog called Germ Blog. I'll put a link to that. And Harley used to be an enterovirologist, and now he does outreach, and he's a physician. He tries to teach people how to prevent transmission of infectious diseases. So he's actually agreed to come on the show, and we'll have him to talk about all these issues. Would these be useful in preventing infection? Let's get it from an expert. Appreciate, Jim, that you sent the email. I know it's hard to do, so I'll tell you later on how I've made it a bit easier for people to send in audio comments. Okay, so that does it for email this week. Next week, I'm going to have a lot more email. We'll try and catch up on all of them. It'll be our monthly email show. And it's time for our science picks of the week. So, Glenn, why don't you go first?
Yes, I am going to highly recommend a book that I finished just actually about a week ago or so. It's a book called Riddled with Life. It's written by Marlene Zook, who is an evolutionary biologist. She's at UC Riverside. I actually got to know Marlene a bit because one of the things I do is I'm the editor for PLOS Pathogens Opinion, the front matter section for opinions, which is an opportunity for scientists to write fairly short and intentionally provocative topics. And I had heard about Marlene's work and asked her to write about gender bias and pathogenesis with various pathogens, including viruses. And that led me to purchase her most recent book, that's Riddled with Life. And I found it to be completely fascinating and really quite charming. It takes a very different strategy from the one we were just talking about, where we look at viruses or other pathogens as the enemy, but rather looks at it from an evolutionary perspective and says, well, pathogens have co-evolved with humans and therefore, to some degree, maybe for better or for worse, have helped to shape who we are. And that, in fact, disease may have been a necessary step to get us evolutionarily to where we are today. So the book is full of fascinating anecdotes. It's clever, it's funny, and it's informative. And I would strongly recommend it. It's in paperback as well, so it's pretty affordable.
I'm looking at it here. Riddled with Life, Friendly Worms, Ladybug Sex, and the Parasites That Make Us Who We Are.
That's right. It's a fairly interesting subtitle, I suspect.
Sounds great. I'm going to order it. And Natalie Angier says, fascinating. That's great. Great pick. Thank you.
Welcome.
My pick is all the virology on the World Wide Web. Virology.net, which is a compendium put together by David Sanders. And he started this years ago, before the web and social media got popular. And it just basically has all the links that you might want to virology. Unfortunately, he doesn't keep it up as well as he should. So I would say to listeners of TWiV, check it out, and if you like it, send him an email and tell him to update it. because I think if he hears from people, he will. And also tell him to add TWiV to the listing, because he hasn't done that. But it's a nice resource, even if it's not updated. A lot of good stuff on there. So that's it. All the virology on the www. Well, that should do it for another TWiV. We're a member of sciencepodcasters.org and promednetwork.com, two other places where you can find great science podcasts. Glenn, thanks so much for joining us. I really appreciate it.
Vincent, it was my pleasure.
I am going to put a link to your website at Fox Chase for people to follow.
Yep, that's perfect. Thanks.
And so go check out what Glenn does, and we'll have him back as well. You can follow TWiV in a number of ways. We have a website, twiv.tv, where you can find show notes and links to all the stories we talk about. Please do send us your questions and comments, twiv@twiv.tv. We love getting them. I know we have a backlog. We'll get to them next week. You guys ask great questions, and they really make us think. And I know I've suggested that you send in audio MP3 files, and now what I've done is I've set up a Skype account for TWiV. It's on Skype. It's called TWiV Podcast, one word, and you can leave a voicemail. You have up to 10 minutes to state your question or comment, and I'll play it here on TWiV. And that means you don't have to do the recording and uploading as Jim did, so it might be a bit easier. So that's on Skype.com, TWiV Podcast, all one word. Call us and leave a message. You've been listening to This Week in Virology, the podcast all about viruses. Thanks for joining us. We will be back again next week. Thanks again, Glenn.
My pleasure.
Another TWiV is viral.